Hello everyone, I am Sandeep. I am Senior Product and Marketing Director for Fapon Biotech. Today I am giving a webinar on recombinant rheumatoid factor. This is a very timely and important topic for the IVD as well as for the pharma industry. Initially, I'll tell you briefly about Fapon. Then we will discuss about rheumatoid factor. Finally, we will take a deep dive into the recombinant rheumatoid factor, followed by a brief overview of our relevant products. Let's first discuss about Fapon. As you all know, Fapon is a leading life science organization. We are in the industry from more than 24 years. We have more than 1300 patents, out of which 500 have already been granted. We are operating in 68 countries and regions worldwide, and we have more than 2500 partners globally. We are at this moment, we are one of the major IVD company specializing in the IVD raw materials. As you can see, Fapon is well recognized internationally, as evident from our multiple awards. We come in the Forbes list in the top 30 international brands. Then we have high EcoVeda sustainability rating, so we are in the bronze category. We are also we have a strong commitment to corporate social responsibility and have the relevant ISOs for that. Most of our growth till date have been driven mainly by innovation. In the first phase, when the company started in 2001, we became one of the leading supplier of antibodies, antigens, and the diagnostic enzymes. Later on, from 2009 onwards, we ventured into core raw materials as well as the reagent services. From 2016 onwards, we started providing the complete solution. Now we are providing the open instruments to the IVD industry, and we also started our operations in pharmaceuticals. And the 2020 onwards, we are building up an ecosystem not only for diagnostics but also for the pharma, and we ventured into the artificial intelligence based solutions for the diagnostics and pharma. So as you can see our journey, we started from China and Southeast Asia, then we ventured into Europe and the US, and now we are working as an ecosystem partner focusing on high specialty applications. We have a global footprint. As you can see, we are already present in 68 countries and regions, and we have 5 sites. Our parent site is in Dongguan in China, which is dedicated to the raw materials, reagents, and the pharmaceutical. Then we have a site in Shenzhen, which is our headquarters for CLIA and the next generation sequencing. Then we have a site in Shanghai, which is dedicated to pharmaceuticals and AI-powered drug development. Then we have 2 sites in the US. One is in Boston, which is dedicated to molecular diagnostic, enzymes, and innovative therapeutics. Then we also have a site in San Francisco, which is dedicated to next generation sequencing, protein sequencing, as well as the AI algorithms. We have a highly specialized infrastructure and very high standards for R&D and production. Overall, we have 100,000 square meter of our facilities. In this slide, you can just get a glimpse of our various activities that we do in R&D and production, ranging from antibody discovery, stable cell line development, high cell density culture, followed by the purification, physicochemical analysis, and the chemiluminescent immunoassays. Fapon has become the most trusted partner to the IVD industry. We have a strict adherence to quality. We have advanced R&D and the production capabilities. We ensure that the supply of the raw materials and the IVD products is stable to the IVD and the pharma industry. We have a comprehensive product portfolio. We have over 1400 IVD products, and every year we have 30 IVD projects ongoing. So we are adding new and new products to our pipeline. And as you can see, Fapon has multiple platforms and the assay formats. So when we develop our raw materials, we ensure our raw materials work for all the multifarious IVD formats that our IVD partners are employing for the development of their products. And we have a very highly experienced technical support and the customer service teams. Now, in the next part, we will discuss about the rheumatoid factor. Rheumatoid factor was a term that was coined in 1939 for the autoantibodies that are directed against the Fc fragment of the denatured IgG. Now, RF is prevalent in 3 different types. RF IgM, it is the most prevalent, like more than 80% of the RF, it is prevalent as IgM subtype. You also have IgA as well as IgG, but these are very minor fractions. In healthy individuals, RF regulates physiological processes. RF IgM are low affinity. They neutralizes the autoantigen, thereby regulating IgG homeostasis. While in case of the rheumatoid arthritis, RF IgM are high affinity, they are monoclonal, and they protects the autoimmunogen from degradation. The formation of the immune complex between RF and denatured IgG promotes the progression of various diseases. RF interferes with several of the lab tests, as is already evident to many of the clinical scientists worldwide. And high serum levels of the RM, it can also interfere with the therapeutic regimen. RF is very important for the diagnosis and prognosis of rheumatoid arthritis, which is an autoimmune and the inflammatory disease affecting 0.5 to 1% of the adults worldwide. It mostly affects women greater than 55 years of age. There is no cure. Annually, around 42 million persons are impacted by this particular disease. Early diagnosis of RA enables more effective clinical management. It slows the progression of rheumatoid arthritis, preventing the subjects from joint destruction, disability, and costly complications. The normal level of RF is less than 20 IU per ml. According to the WHO, less than 25 IU per ml are considered negative, while the RF levels ranging from 26 to 75 IU per ml are considered low titer positive, and greater than 75 IU per ml are considered high titer positive. Now, RF testing is very important for rheumatoid arthritis. The sensitivity of RF is between 60 to 90% for the rheumatoid arthritis, while the specificity is around 85%. But the combination of RF with ACPA enables very high sensitivity. The determination of RF is very important in case of the autoimmune diseases and inflammatory or the infectious diseases such as tuberculosis and hepatitis. Further, it is important to determine the RF levels to predict the therapeutic response. So this slide shows like the pathophysiology of the rheumatoid factor. On the left side, when the person is healthy, then RF has a major role. So it is low affinity, it is polyreactive, and it can effectively counteract the autoantigen. So you have the IgG homeostasis. But when you, on the right side, when you have the rheumatoid arthritis, your RF IgM, they turn into monoreactive, they are high affinity, and they protects the autoantigen. This results in accumulation of autoantibodies, and then most of the complications starts in the patients. On the right side, you see the pathophysiological mechanism, how it works. From the B cells, you have the rheumatoid factor, and then the rheumatoid factor forms an immune complex with the IgG. RF may acquire pathogenicity by forming the immune complex with the ACPA IgG antibodies, and this causes inflammation by stimulating the secretion of pro-inflammatory cytokines. In this particular slide, we discuss about the clinical pathophysiology of RF. In this slide, we discuss about the clinical pathophysiology of rheumatoid factor. The formation of the immune complex between RF and IgG at the sites of synovial infiltration. It leads to the activation of the complement system, leukocyte infiltration, and increased inflammation. Now the RF epitopes, they are located in the Fc region of the IgG. So they are either in the CH2 or the CH3 regions towards the inside. The binding of the antibodies to the pathogen surfaces induces the conformational change, which exposes the Fc region and the epitopes of rheumatoid factor. Certain other physicochemical treatments such as heat treatment at 57 degrees centigrade or the physical adsorption on a hydrophobic surface, they also make the RF epitopes exposed. So technically, whenever there is denaturation of the IgG, the epitopes of rheumatoid factor in the Fc region of the denatured IgG, they are exposed. Now the detection of the rheumatoid factor is done by several techniques, but the most used is latex enhanced immunoturbidimetry. The reason is it is highly sensitive, it is quantitative, and it is high throughput. Let's see how native RF is produced. Native RF is extracted from disease state plasma of rheumatoid arthritis patients. So it contains all the naturally occurring autoantibody variants. However, there are strong limitations. The major limitations are it is expensive, it has low yield, the lot-to-lot variability is very high, and there is a risk of continuous supply. So technically, it is not highly sustainable model. And the native sera from the patients, they might contain several infectious agents. So you have to handle the native sera in the biosafety level 2 facility. So technically for the industry, it is much better to employ the recombinant rheumatoid factor. Now recombinant rheumatoid factor, it is developed by a recombinant expression systems. It is highly sustainable because it is low cost and continuous supply is assured. Then the lot-to-lot reproducibility is very high. Then there is high yield. There is no risk of blood-borne infections, so you don't require very costly BSL level 2 infrastructure. Further, it is highly stable, so it has improved shelf life, and you can further optimize the buffer formulations. However, the raw material developers, they have to ensure that the recombinant RF is stable. It is eliciting the same response as you are getting from the native rheumatoid factor. The lot-to-lot reproducibility needs to be very high, and it needs to be highly specific. Recombinant rheumatoid factor has different applications. It is used as positive control and calibrator for various assays, and it is also used as a reference standard. This slide provides a glimpse of how recombinant RF is produced. The process involves the immunization of the particular animal species with the immunogens. This is then followed by different antibody production schemes. At Fapon, we are using the single B cell technology. This is highly sophisticated production scheme. So initially we do the single B cell sorting. This is followed by the gene sequencing, expression, and the vector. Then we have the Chinese hamster ovary cell engineering, which is followed by the fermentation, expression, purification, quality control, followed by the release of the recombinant rheumatoid factor. Now, commercially, most of the IVD manufacturers, they already know that there is a strong requirement for the animal origin free products. So the recombinant rheumatoid factor that we are providing to the industry, it is animal origin free. Animal origin free means the product does not contain any primary or secondary raw materials which are derived from tissue or the body fluids of the animals. This is strongly emphasized by US FDA as well as the EU authorities. And many of the IVD manufacturers, they have a strong preference for animal origin free raw materials to comply with the corporate social responsibility. Fapon's recombinant RF production capacity is very high. We can produce ranging from 0.8 to 60 liters in a single batch. So let's say if some IVD manufacturer require up to 360 liter of recombinant RF, we can produce that particular batch in one go. And we have very high lot-to-lot reproducibility. So we can provide the recombinant RF to the IVD manufacturers immediately because we always have it in stock. But if you have a very high requirement, then we have a particular production batch every quarter, and we can produce the very high quantity as per the customer requirement. So Fapon is offering two types of the products. One is the native RF, but we recommend our clients to go for recombinant RF because it is highly stable. The lot-to-lot reproducibility is very high. It is low cost, and we ensures a continuous supply. Here you can see for the natural, we provide an assurance that you will get 900 to 1100 IU per ml from a single batch. But in case of the recombinant RF, we assure the clients they will be getting higher than 2000 IU per ml. So when we do our QC testing, this is the particular setup for our QC testing. As I mentioned, for the native we have 900 to 1100 IU per ml, but for the recombinant we assure higher than 2000 IU per ml. Here we did the direct comparison of our recombinant RF with the native RF in the range from 15 to 150 IU per ml. As you know, like within the pathophysiological range, that is higher than 25 IU per ml. We don't have any significant difference between the native and the recombinant, so the variability is less than 10%. When you go near the limit of detection, that is at 15 IU per ml, so we have around 19.9% variability, which is expected as per the CLSI norm, because when you go near the limit of detection, you have higher matrix effect, but it is still within the acceptable standard. Now, these results are based on the linear dilution of our recombinant RF, but when you employ it in the actual matrices of your calibrators and controls, that is depleted charcoal stripped serum or plasma, then you will get much better performance. Here you can see the direct comparison of the stability. The recombinant RF is highly stable at 4 degrees centigrade, at 37 degrees centigrade. Further, the freeze thaw stability is also very high. So we recommend the IVD manufacturers to go for the recombinant rheumatoid factor. Here you can see the lot-to-lot reproducibility of the 3 different batches. It is less than 10%. And as we already discussed, we assure higher than 2000 IU per ml for all our manufacturing batches for recombinant RF. In this slide, we did a direct comparison of our recombinant RF with one of the competitors in the market. So we have a very good correlation with the on-market products. Now let's discuss about some other relevant products that could be of use to the IVD manufacturers. Similar to the recombinant RF, we have raw materials for quality controls and the calibrators for a wide variety of the biochemistry assay. Here you can see the complete list. And very soon we will also be launching the full range CRP. Additionally, we have raw materials for calibrators and controls for immunoassays. our infectious disease as well as like the torch panel. It is the most used in the IVD industry. As you can see, we are providing the humanized antibody for the torch panel. Humanized antibody has strategic advantage that it provides the same immune response in the human body. Now, in case of the humanized antibody, the FC region comes from the humans. Apart from the antibodies, we also have antigens. These are our antigen raw materials for QCs and calibrators for immunoassay. Lastly, Fapon also have very attractive product line for heterophilic antibody interference blockers. Our heterophilic antibody blockers, they can effectively block human anti-mouse antibody as well as the rheumatoid factor. These specific blockers are produced using human IgM or RF as your immunogens. Then we employ the single B cell technology. These specific blockers, they can take out the interference due to HAMA and RF in your assays. This is a list of our heterophilic antibody blockers. As you can see, most of our blockers, they work via passive as well as the active mechanisms. That means they prevent the entry of the heterophilic antibodies into the specific reaction system, and they also actively bind to the heterophilic antibody. This combined mechanisms effectively takes out the interference due to HAMA and rheumatoid factor. And our blockers, we have already checked on multifarious assay formats. That is for chemiluminescent immunoassay, for ELISA, for rapid test, for immunoturbidimetry. So the IVD manufacturers, they can use it for multiple formats. Thank you everyone. I hope I have provided you good insights into recombinant rheumatoid factor. If you want to know more about our recombinant rheumatoid factor and our product portfolio, you're most welcome to visit us at ADLM at our exhibition booth 3816. We are looking forward to meeting you.